My name is Rabbi Yaakov Spivak. Welcome to the session on stem cell research, which I've been asked to chair. I would basically like to introduce the two speakers. That's my job and responsibility. This opening session on the possibility of, quote, creating life has always been fascinating in Jewish literature. As far back as the Gemara in Sanhedrin Samach Hey, which talks about, and we don't know what it means, it talks about the fact that Rava actually created a human being that he could talk to, but he could not respond. What the sages meant, I'm not totally sure, but the subject of being involved in creating life has always fascinated people, and today, based on the scientific research that we're going to hear about, we're going to see how it's going to get into the hands of not only the Ravas of the world, but of many, many more people. The first speaker is Dr. Miriam H. Furman. Dr. Furman was trained as a molecular biologist, first receiving an undergraduate degree in biochemistry and cell biology from the University of California, San Diego, followed by a PhD in biological studies from the University of California, Irvine, under the supervision of Dr. Hung Fan. The subject of her thesis work was the determinants of the disease in acutely transforming retroviruses. As a postdoctoral fellow, she studied the regulation of the alpha fetoprotein gene during development with Dr. Shirley Tilghman at Princeton University. In 1990, Dr. Furman joined the biochemistry department at Downstate Medical Center as an assistant professor. As a member of this department, her teaching responsibilities in the medical school include directing the Genes to Cells block of the first-year curriculum, where she teaches molecular biology and genetics. At Downstate, Dr. Furman started her studies of the genetic regulation of gene expression during liver regeneration. The ultimate goal of her laboratory is to identify genes regulating liver regeneration that may be involved in preventing liver carcinogenesis. She used a biochemical and genetic approach to this problem, utilizing and contributing to the data of the mouse and human genome projects. Identification of such genes may provide information in the future for more accurate diagnosis or therapeutic targets for development of new drugs with greater efficacy and fewer side effects. Funding for this work has been provided by the National Institutes of Health, National Cancer Institute grants, etc., which you can read in your in the catalog provided. She has been invited to present this work at international meetings at Cold Spring Harbor, New York, Snowmass, Colorado, Orvieto, Italy, and Kyoto, Japan. It is my distinct honor to present to you, ladies and gentlemen, Dr. Miriam Furman.
Thank you for such an introduction. I'm unaccustomed. Okay. So you guys can see it, I can't. No, I can't see anything at all. Okay, well, if you'll pardon me, I'm going to have to fix this on this big screen. I know it takes away all of this. No, it takes away all the magic, but you got to do what you got to do. To be honest, I haven't had this happen before. How about if we detect the displays? Bingo. Oh, now you can't see anything. Now you can't. Good. This is always one of those, I don't want to say nightmares, but one of the things that you fear when using technology is that for some reason it's not going to work. Well, look at it this way. Now that this technology hasn't worked, that should be the end of the glitches for the day. So I've, given up the magic of my presentation such that everybody else's presentation should work very well. So I've been asked to present the scientific background on embryo and stem cell research. As indicated by the rabbi, I don't work on this. However, I've done some research, and like most scientists, I find the topic very fascinating. Embryo and stem cell research is not just of scientific interest. First of all of you are here, and there are potential medical discoveries that will aid in the treatment of disease. But also it's a matter of public interest. In the past couple of weeks alone, there have been at least 2 references to cloning and stem cell research in the popular media, in TV dramas of all things. And on August 9th of 2001, stem cell research was the subject of a presidential address. So I'm going to tell you a little bit about stem cell research. I'm going to tell you a little bit about what is happening now, and hopefully some of the potential therapies that will be developed from this research. So on this slide, I've listed the uses, or what are thought to be some of the uses. In many cases, I guess in all cases, this technology is under development. So in fact, we don't know that these are going to be the only uses, and we don't know that these are going to be the uses for these cells. But it is highly likely, and in fact it has already occurred, that stem cells have been used in the study of human development. It is anticipated that having these stem cell lines will permit people to test new drugs that have been developed. And finally, and probably most excitingly for all of us as human beings and you as doctors, is that it may open the door to cell-based therapies. For example, we already use bone marrow transplant. That has been in the literature for many, many years, and in fact it's in the clinic. But it is also hoped that you may be able to repair heart damage by the injection of cells as opposed to the transplantation of a new heart. It is also hoped that neurological damage may be repaired by the injection of these cells. And it may happen first for spinal injury, allowing the spinal cord to be reconnected. And then one of the more obvious potential uses is in the treatment of of burn victims. It is hoped that these cells may permit us to grow new skin to help people recover from very severe burns. So what are stem cells? Stem cells, by definition, they are omnipotent. That is, they can divide and produce more stem cells. So they have unlimited renewal. This means that they can be grown in great quantities. The cells themselves are unspecialized in that they do not express genes or they do not carry out functions that are specific for given tissues in the body. However, they can give rise to specialized cells, and in my very crude PowerPoint drawings, I've tried to indicate muscle cells, nerve cells, and columnar cells of the intestine. But let me give you an example of how stem cells can be treated such that you can, in fact, spontaneously or not so spontaneously, allow the development of cells with specialized functions. So what you can do is take these cells and allow them to differentiate in their little dish into something called an embryoid body. If you then select these cells that express a certain gene and then treat them with things like basic fibroblast growth factor and laminin, you can then select out cells that are going to be of the neural lineage. By carrying out this procedure, you can actually cultivate cells that secrete dopamine and serotonin. So how do you make stem cells? This slide makes it look very easy, but in fact, it is, I guess there are very subtle parameters that play into each of these steps. You start out with a fertilized egg. And in the fertilized egg, if you get development of a zygote and an embryo, you can get the development of these cells, and they're from the inner cell mass. I'll show you some more realistic pictures, if you will. These cells can be removed from this developing blastocyst and put into a culture dish. From this culture dish, they can be allowed to grow by providing them with all the nutrients that they need. And usually there's a feeder cell line. In other words, these cells are not generally plated on plastic alone, but rather the bottom of the dish is coated with cells that are already growing, and they are generally mouse embryo fibroblast cells. And this is important to consider when thinking about using these cells in therapeutic uses. And it is true that this embryo, if you will, gives its all to become a stem cell line. So here's my more realistic pictures of what we're talking about. Here's the fertilized egg. And to be honest, I got these pictures from the New York Times website, so you could go check them out yourself. So here's the fertilized egg. At day 4, you get the formation of a blastocyst, and as part of the blastocyst, you have the cavity, and then it's up at one end, you have what is called the inner cell mass. A day later, the inner cell mass forms this little ball of cells, which I've tried to show at higher mag in this picture here. The zygotes for these cell lines are typically obtained from fertility clinics. So these are leftover embryos that the couples have decided would not be used. Furthermore, they have generally specified that they should be destroyed if they're not going to be used. However, they can be donated to people who are doing this research, or before August of 2001, they could be donated. To give you an idea how early in development this is, here's what happens at day 14. Day 14 is the time when defective embryos will generally spontaneously abort. So generally, at least before the advent of home pregnancy tests, people did not, or women, did not know that they were pregnant at day 14. And defective embryos would spontaneously abort if, as I said, if they are defective. It is with the advent of home pregnancy tests that women can tell essentially if they're, I think 9 days pregnant is the most sensitive one. So if you know that you're pregnant at 9 days and then you are not pregnant at 14 days, you know that you have been pregnant. And generally it's because the embryo has been, is defective. To give you a further signpost, if you will, I've included some pictures and the times that they appear for so-called recognizable structures. So at day 33 of gestation, you have the formation of a limb bud. And at day 37 of gestation, you have the formation of an eye, and you can sort of see the umbilical cord. So these procedures take place very early on in development. As you, I'm sure, well know, this has been a source of controversy. So people have looked in other places for cells that can accomplish the same results. So you may have heard about research involving so-called adult stem cells, or perhaps more accurately, stem cells that are found in adult tissue. So let's talk a little bit about stem cells that are found in adult tissue. It is generally thought that they exist as a reserve for repair or renewal of that tissue. The tissue that I work on, the liver, it is thought that it does have stem cells, although it is not required for liver regeneration. And I could go on about that at great length, which I'm sure you're not very interested in at this point. Anyway, it is well known that there are hemopoietic stem cells and bone marrow stem cells. So these are actually cells in the bone marrow that support the hemopoietic development. There have been identified neural stem cells. Similarly, epithelial cells, stem cells, and skin stem cells. I could go into, well, why don't I tell you a few of the cells that come from these. As you well know, I'm sure, hemopoietic stem cells can be caused to differentiate into red blood cells, lymphocytes, natural killer cells, the cells of the immune system. Neural cells, they've found neural stem cells, and they can induce differentiation into neurons, astrocytes, and oligodendrocytes. The GI tract has a fairly well-characterized stem cell compartment, and these cells are required for the replenishment of several types of cells, including absorptive cells, goblet cells, Paneth cells, and enteroendocrine cells. And then skin stem cells can give rise to keratinocytes and follicular cells, that give rise to the hair follicle and the epidermis. So there are stem cells that are found in adults. As with fetal stem cells or embryonic stem cells, there are many, many unanswered questions about adult stem cells. For example, was the list that I gave you of tissues where stem cells could be found exhaustive, or are there other stem cells that can be identified in other tissues? Can these cells be extracted? In the case of the bone marrow, since this is done, we know that you can extract bone marrow stem cells and likely hemopoietic stem cells. Actually, you can extract hemopoietic stem cells without threatening the life of the individual, of the donor. Can adult stem cells be propagated? And just how plastic are these cells? In other words, if you have bone marrow stem cells, can you induce them to differentiate into other useful cells where those stem cells might be harder to get at? For example, in the brain, if you need new neurons, it is unlikely that you want to isolate stem cells from the individual's brain to repopulate their brain because of the damage. Now, a while back, and I can't tell you exactly how long, I think it was about 2 years ago, something called transdifferentiation was reported. That is, bone marrow stem cells were reported to differentiate into other cells, including neurons, muscle cells, and liver cells. New reports suggest that this is an unusual and very rare phenomenon. In fact, in Science of September of this year, Irv Weissman, who is an expert at stem cells, hemopoietic stem cells, from either Stanford or UCSF, and I can't remember which, made chimeric mice to look for this transdifferentiation. And he looked at a total of 15 million cells, and he detected no transdifferentiation. He also did another experiment in hopes of looking for transdifferentiation, and he looked at another 12 million cells. I'm just trying to impress you with the numbers. And found no transdifferentiation. So from his data, one can infer that transdifferentiation is A, difficult, and B, rare. It doesn't mean that we can't induce it in culture. So what are these elusive properties of stem cells? Well, first of all, you need long-term self-renewal. And this is important for maintaining the cell lines, but also for generating a large number of these stem cells. They have a characteristic pattern of gene expression. An example of a marker that can be used to identify stem cells, very primitive stem cells, is called OCT4. They need to have a normal karyotype. And finally, they need to be pluripotent or omnipotent. They need to be able to differentiate into many, many types of cells. And this is generally tested by looking for spontaneous differentiation in culture, by inducing differentiation into specific cell fates, by manipulating the culture conditions, and by injecting cells into immunocompromised mice to see if they form a type of tumor called a teratoma. It is known that teratomas develop from very primordial stem cells. So let me compare fetal and adult stem cells. Fetal stem cells are pluripotent. They can form many more tissues than the multipotent adult stem cells that have been identified. Fetal stem cells are readily expandable in culture. So if you start with a few, under certain parameters, you can grow a lot of them. Adult stem cells can be cultivated, but there's much more limited growth. Now, if you generate a fetal stem cell line and you want to use it to repair large numbers of people's tissues, remember that the fetal stem cells have all the immunological markers from the donor. So they may be rejected upon transplantation. Adult stem cells could potentially be isolated from a patient, caused to differentiate or caused to form the tissue of interest, and then re-injected or replaced in that patient. So it would be immunologically the same as that patient, reducing the possibility of rejection. Now, this is what is currently known. Whether this will change with time is unknown, and I suspect that it will. In either case, both fetal or adult stem cells, the amount that we know is far outpaced by the amount we don't know. So let's talk about some of their potential uses. In research, it is thought that stem cells will help us understand more specifically some of the events of human development. Obviously, if you're a molecular biologist, it is very difficult to study human development. However, these stem cells may maintain enough characteristics of the whole organism that will allow it to be studied. Cancer studies will also be aided by these lines. And that is because stem cells may maintain or can be differentiated in culture and may maintain a lot of the control characteristics of normal cells as opposed to the cell lines that we currently work with in the lab. The cell lines that we currently work with in the lab are generally derived from tumors and as such have lost a lot of the characteristics that control growth. One of the biggest hopes, of course, is that stem cells will be useful in therapeutics. The diseases that are most commonly mentioned are diabetes, Parkinson's, Alzheimer's, and as I talked about before, perhaps we may be able to fix damaged spinal cords by the use of these cells. I don't know how much you've heard about this, but Nancy Reagan has become quite the advocate for fetal stem cell research because of her husband, Ronald Reagan, who was president, now has been diagnosed with Alzheimer's. I find this particularly interesting because I would put down money that would say if Ronald Reagan as president could have supported stem cell research, he would not have. I would bet money on that. So what are the current guidelines on stem cell research? Well, on August 9th, 2001, President Bush announced that federally funded research on embryonic stem cells could go on only on existing cell lines. And according to the data behind the initial announcement, they reported the existence of 64 cell lines. However, that number was then corrected or revised to be 24 or 25 stem cell lines. That was what was thought. Currently, there are now about 7 lines in U.S. academic institutions and 13 lines in U.S. companies. The rest of these lines are found outside the country. This includes 19 cell lines in Gothenburg. And further investigation of these lines in Gothenburg says that these lines are not really lines at all, but rather new isolates. The actual number that the scientists in Gothenburg expect to derive is 3 to 4 cell lines. Currently, or I should say as of August 14th, 2002, it is thought that there are 78 eligible stem cell lines. And that is, what does it mean to be eligible? That work was started before August 9th, 2001. Of these 78 stem cell lines, about 16 are available for distribution. Now, what does it mean to be eligible? So first, as I mentioned, they must be created from embryos that were created for reproductive purposes that were no longer needed. Informed consent must have been obtained for each donation. And finally, there must not have been any financial inducements for this donation. I do not know if that includes tax deductions. So what are the current lines that are available? Well, they all require feeder cell lines just to maintain life of those lines. By definition, if you have a limited number of stem cell lines, you have limited genetic diversity. Remember, the genetic complement of each of these stem cell lines reflects the donor. And finally, the quality of these embryos may be limited by the conditions of the donors. Remember, these were created in clinics to assist in reproduction. These are people who've had a hard time conceiving on their own, probably because the mothers are are a little bit older, maybe they're 35 instead of 20. And what is the reason for this difficulty in conception? Is it genetic? Could it be compromising the quality of the genetic material in these stem cell lines? In any event, it is certainly limited. So now to switch gears a little bit, I wanted to talk a little bit about cloning. Although if you talk to the people who work on it, they don't want you to call it cloning. They want you to call it somatic cell nuclear transfer, which explains why people call it cloning. But the term cloning evokes images of science fiction. This was actually the cover of a New York Times magazine that came out, is the date on it? It looks like February of 2001. It evokes images of The Boys from Brazil, the movie Sleeper, where they wanted to clone the nose. What you have to remember is what a clone is. It is a younger individual, whether it's animal or, God forbid, human, with the same genetic complement as the donor cell. It is not a younger you. There are other influences in development, okay? If we were to clone humans. It's not a young, Dolly the sheep is not a younger version of her, I don't know if to say mother or father. Now people, when they talk about cloning, they differentiate between reproductive cloning and therapeutic cloning. And as far as I am aware, there is no reputable scientist who's talking about using cloning for reproductive services. There are a couple of scientists, I believe one has recently moved to Italy, to use cloning for reproduction. To be honest, if Howard Hughes were alive today, I'm sure he'd be trying to clone himself with his toenails that he's saved. And we've also heard about the Raelians who are trying to clone their leader. But these are, well, I don't have to tell you what these are. So let me tell you a little bit about the procedure. It sounds, again, straightforward. In practice, it is not. You remove the nucleus from an egg. You culture somatic cells from the individual that you wish to clone. So remember, when you remove the nucleus from the egg, yes, you are removing the genetic material of the nucleus, but there are also genomes found in the mitochondria that are in the cytoplasm. You then transfer the nucleus from that somatic cell into the egg, and you coax it to develop. If you transfer these developing zygotes to a uterus, it's called reproductive cloning. If you take these blastocysts and put them in a petri dish, then it is considered therapeutic cloning. So there are some legitimate uses for human somatic cell nuclear transfer, or thought to be legitimate uses. One is that you will be able to study the genetic contributions of disease. Remember, if you're dealing with stem cell lines, fetal stem cell lines, their genetic complement is determined by the donor. So we have, in theory, 78 to choose from. You can clone, if you will, anybody or any animal. You can use these cells to study how drugs may function in cells with genetic complements. Again, looking at the diversity of humankind. More in the future, people are talking about donor cells and donor organs. Again, you can grow a specific cell type, induce it to differentiate, and perhaps grow a liver in a petri dish for an individual who might need a new liver. As I said, it looks straightforward, but in practice it is not. Attempts to clone have been successful in sheep, cattle, mice, pigs, goats, rabbits, and cats. Many attempts have been still unsuccessful in rats, rhesus monkey, and dog. The exact requirements for success in cloning seem to vary with a given species. Interestingly, companies are getting into the idea of cloning because they want to clone pets for people. I'm sure agriculture is interested in cloning. And something to me is very interesting is on this list, I don't see horses. And given the interest in thoroughbreds, I'm really surprised that horses aren't on this list. So what do I mean by successful? Successful is not that successful. Overall, cloning has a success rate of less than 4%. So realize that a lot of zygotes die. There's very high fetal, perinatal, and neonatal loss in this process. Some of the things that have been documented are abnormal gestation. The placenta has vascular reduction, and there's an accumulation of fluid in the hydroallantois. So there are lots of things that are going wrong. In offspring that are carried to term within 24 hours of birth, it is noted that there's a high proportion of respiratory distress, an increased birth weight, and cardiovascular abnormalities. Later on, it is noted that a lot of these offspring suffer from so-called large offspring syndrome. They're just too big. There's a frequent immune system failure, and there have been noted structural abnormalities of the brain. It is thought that imprinted genes, genes that are expressed differently depending on the parent from which they are inherited, is responsible for some of these difficulties. As I said, there are some so-called legitimate uses for cloned cells anyway. Testing of drugs, cell replacement, and organ replacement. Again, we do many, many transplants now, but there are not nearly enough organs to go around for everybody who needs a transplant. So I would just like to sort of summarize here. And the future of embryo and stem cell technology is really just beginning. There are many research uses that are only now beginning to be realized. There will be therapeutic uses if this research proceeds in the fashion that everybody expects. And the biggest thing that I can tell you is that there's so much that we don't know that all the lists of things that I've told you may turn out to be wrong 10 to 20 years from now. Things may develop in unexpected ways, as seems to be the way of science. So I'll stop there. Should I take questions? Are there any questions?
Are there any questions for Dr. Furman?
Yeah.
Cord blood stem cells, are they more like fetal or adult? How do they function? I know there's a big industry now.
There is. You can, when a child is born, you can save the cord blood. To be honest, I don't know. I should have looked that up. My apologies. I don't know. Yeah.
Yes, sir.
What are some of the risks of implanting stem cells?
I'm sorry?
What are some of the risks and complications in animal birth? Once you implant the stem cells, any complications or risks of death when you get cure anything in the process of implanting?
So this is for therapies as opposed to reproduction. There's always the potential for immunologic rejection.
Let's say you use an adult stem cell, one from that hurt.
Well, right, so if you use a fetal stem cell that you've induced to differentiate in culture, its nuclear genome is from the donor. So if the immunological complement of the donor differs from the animal that you are trying to transplant it into, you're of course going to have immunological rejection. I believe with some of the Parkinson's experiments, there's been a problem of the cells being too active. So they've secreted way too much dopamine and have caused other rejection or other problems in treating. I believe that actually happened in people. They did that study, and I think they halted stem cell transplants into Parkinson's patients. Now that happened a couple of years ago. I don't know what the further developments of that are.
That's the final question.
Okay.
Thank you very much, Dr. Furman. I am reminded of a story which is told about the Lubavitcher Rebbe during 1977, he had a heart attack, and he told his doctors that he had read about the fact that the same, in the fetus, the same stem cells that would create scar tissue also can create normal muscle. And he had hoped that one day that some of this research would bring a science to be able to create normal muscle in the heart after a myocardial infarction.